Good morning. Welcome back to Track 3, Drug Development, Getting into the Clinic, sponsored by PRA Health Sciences. We hope you enjoyed your morning break, sponsored by AMD Serrano, and are ready to dive into our first breakout session. My name is Angie Rowe, and I will be your host for these morning sessions. Once again, we would like to remind everyone that we are live streaming, and I would like to welcome our remote attendees to the session. As a reminder, we have our mobile app available. Be sure to download it to use some of our highlighted features like live Q&A. And without further delay, I would like to introduce our first session of the day, Getting from the Lab to the Clinic Faster, Advancing Translational Science, presented by Christopher Austin, Director of the National Center for Advancing Translational Sciences at the National Institute of Health. Please join me in welcoming him.
So you've already welcomed me once, so we don't have to welcome again. What I'm going to try to do is put some meat on the bones of what John Crowley and I talked about this morning, and really what I'm going to focus on is opportunities that I think you might be interested in, and how you actually get involved to take advantage of some of the things that I'm going to tell you. And I'm going to give you, there's a slide at the end that is a screenshot of something that should be in the mobile app from this session that should list descriptions of the programs that NCATS has, what they do, what the entry criteria and the exit criteria are, and then who the program contact is. If it should be in there. If it's not, then we can get it fixed. But it should be in there. Okay. So I want you to know how I describe this to policymakers in Washington and anybody else who will listen. That is, why are we in this pickle? And I mentioned, and I think you all know the pickle we're talking about. It's not that this problem is new. I mean, the issue of undiagnosed or untreatable diseases and trying to develop treatments for them has been around for as long as human beings have, and healers trying to do something about them. The difference is in how much opportunity there is now. And how much recognized need there is now, which simply did not exist even 30 years ago. So on the left side here, what I'm trying to get across is the left side is how much we know about ourselves in health and disease. Genome Project, that thing on the bottom is some induced pluripotent stem cells that have been differentiated into neurons and their nerve cells in their growing processes. And I have no notes. I'm not sure why that is. What did I do? Ooh, morbid scorecard. What it's doing. Somebody in the back, it looks like it's showing the notes field instead of the, looks like somebody hit the wrong button back there. There we are. But what's on the right side are some examples of the fact that all this knowledge on the left side really hasn't made it to patients. On the right side, there's an Institute of Medicine report that came out a few years ago called Shorter Lives, Poorer Health, that sort of says it all. And the picture on the bottom is a boy with progeria, it's a rare premature aging syndrome. And the problem here is that we just don't go very well around that virtuous circle that John and I were talking about before, or another way to put it is to go from the left side to the right side. And so there's enormous opportunity and need that we think are coming together now, and we can do something about that. So to just graphically illustrate the issue that we're in, and you all live this every day, because you'll be in this graph somewhere probably, is the number of human conditions, most of them which are diseases, the genetic or molecular basis, scientific basis of which is understood. So when I graduated from medical school in the mid-'80s, that number was less than a dozen less than a dozen human diseases, the molecular basis or genetic basis of which we understood. And this is what's happened since then. In the ensuing 30 years, that number has gone from less than a dozen to now about 6,000. Most of these are rare diseases, of course. And all this is cause for celebration, but this is not cause for celebration. That is, if you add up the rare diseases and common diseases, how many of these diseases have treatment? That number is about 500. And that number increases by about 3 or 4 a year. So you can, if you do the math then, if it increases by 3 or 4 a year, there's 6,000 which have untreated, which are currently untreatable. It gets you to the 2,000 years number that I quoted before. And you might say, well, gosh, you know, we all know that it takes 15 years or so, as John mentioned, to develop a drug. And so maybe all we have to do is wait. And we will see a similar increase in the development of drugs. And unfortunately, you would be wrong. And you'd be wrong because of a contrast of two graphs I'm now going to show you. This is a graph you all know about, whether you know about it or not. Moore's law is the observation in the computer industry that the number of transistors you can fit on a microprocessor has increased linearly, and the cost has decreased linearly since the 70s. That's why what was a supercomputer in the 70s you can now fit into your iPhone. So this dramatic improvement in efficiency and decrease in cost, otherwise known in economic circles as productivity, has great increase in productivity, and we see it everywhere around us. At the same time, however, you have to ask, well, what's happened in therapeutic development? Well, this has happened in therapeutic development. This is a slightly apocryphal name, Eroom's law. This is Moore's law spelled backwards. This comes from a paper in a scientific journal called Nature Reviews Drug Discovery a couple of years ago. And it looks at the productivity growth in therapeutic development during the same period of time, in this case from the 50s. What you're looking at here is the number of new drugs approved by FDA per $1 billion spent. So if any of you are in business, you know the whole idea is that you're going to be able to make more widgets for the same amount of money. So the cost of your project, cost of your good comes down over time. So your productivity increases. What you're seeing here is dramatically negative productivity growth in developing therapeutics over the last 60 years. And this is, for those of you who are technologically inclined, you might notice this is what's known as a logarithmic plot. So it actually makes the slope look less bad than it otherwise would be. And there's a couple of things I want you to notice about this, that this has happened. This slope, this trend has not been affected by any of the epical changes in science and medicine that we all know about in the last 60 years. So invention of computers, recombinant DNA, CAT scans, MRIs, the Genome Project, cloning, gene therapy, editing, you name it. None of them has had any effect. As a matter of fact, there are some people who argue that all of that information has actually confused people so much it's made matters worse, not better. But in any case, the trends are not positive, right? So we can't just keep doing what we're doing and hoping that things are going to improve. I actually looked in a particularly sort of pessimistic moment. I asked, well, gosh, wouldn't it be nice to know when this graph goes to zero? And actually, that number, it goes to zero. It can never officially go to zero because it's a log plot, but it goes to zero in about 2050, which means that officially, unless something changes, there will be no more drugs. Or in other words, the cost of a new drug will be infinity, which is pretty much what it is now. So just imagine, and this is important for all of us who deal with prices of rare disease drugs these days. Think about any industry where over 60 years, the efficiency of making the product goes down and down and down and down. What would happen to that industry? They would acquire each other, they would go out of business, and the products that they do make would be exorbitantly expensive. And this is true whether they're drugs or ball bearings or shoes or televisions or anything else, and that's exactly what you see. So this is the problem that NCATS, among other things, the things that NCATS was started to to solve. And it was clear to us at the very beginning that the problems we're dealing with here are not subtle. They are not going to be solved by a 10% improvement or a 20% improvement or by hiring McKinsey to come in and optimize our system. This is not even close. We need to look for changes which will result in logarithmic improvements, exponential improvements, 10-fold, 100-fold improvements. And where this comes from is the kind of thing that you were hearing about on the last panel, for instance, from Transparency. Completely different ways of thinking about this process that we're involved in. So that's really what NCATS is all about. New ways of doing it, new methods, new technologies, new paradigms, new collaborative structures that will get more treatments to more people more quickly. So before being in this space, it's important for you all to understand how we define the word translation. It's unfortunately that we have a name like this in our title because we are the only institute or center of the 27 at NIH that has a word that means in its name that has a meaning outside of biomedicine. So for instance, my colleagues who work at the National Cancer Institute, pretty much everybody knows. You tell somebody you work at the National Cancer Institute, they know what you do because they know what cancer is. If you say that you work at the National Center for Advancing Translational Sciences, what is that? I get asked not infrequently, you guys do linguistics? Is that what you do? Because that's what translation means to a normal person. So what does translation mean in the biomedical context? It means what you all are trying to make happen every single day. That is the process of turning what appears to be a promising observation that somebody makes for the first time, could be a scientist in the lab, could be a doc in the clinic, could be a community health worker, makes an observation and a light bulb goes off in their head and they say, aha, this observation tells me something which I never knew before. If I can reverse this, then maybe it will reverse the process in front of me, in this case, the disease. And that means anything from a diagnostic to a therapeutic to a medical procedure, et cetera. So what is the field of translational science if we're advancing translational science? It's the field of investigation that seeks to understand what the general principles are by which this process happens. With the hypothesis that if we understand what we're doing here, if there really is a book, a methodology book that tells us how to do this and what's likely to work and what isn't, then we'll be more successful. And that's a general observation in every area of human endeavor for a variety of reasons that has never applied to this whole process of translation. And that's an interesting separate discussion about why that happened, but you live it every day. I have these conversations with parents every day where they say, well, gosh, when my child was diagnosed or I was diagnosed, I went to the doctor and I was astounded to find that not only does nobody have any idea what this thing is, what this disease is, but there's no treatment for it. As a matter of fact, nobody's working on a treatment for it. And the likelihood of there being anytime soon isn't very great. And you say, how can that possibly be? And it's because there has never been a systematic approach to this until the last 5 years when NCATS was founded. Okay, so I'm not going to go through these, and you'll be glad to hear this, but people sometimes want to know, well, so what do you guys actually do? If you don't work on a particular disease, if you're not trying to cure cancer or cure something else, what do you guys do? Well, this is a list that you may have heard either yourselves or colleagues talk about. These are the things that bedevil every therapeutic development project, going from the basic lab to people getting better due to an intervention that's been developed. It doesn't matter whether you're working on Alport syndrome or eczema or Alzheimer's disease, it doesn't matter. They're all the same problems. It's one of the reasons that they've never been worked on, because it's been nobody's day job to solve them. So we work on all of these areas. And as I said to John, the way we do this is to develop new ways of doing all of these things. And then we do demonstration cases. We do hundreds of individual projects on individual diseases, particularly on rare diseases, as ways to figure out whether we're actually doing these things better, faster, cheaper. It's important, though, and again, this is coals to Newcastle for this group, that the problems in translation are not just scientific. There are huge scientific issues and really interesting, frankly, and important scientific issues in this space. But it's unlike other areas of science. In translation, you can get all the science right and still fail. And you fail for what might be called soft science or social science reasons, or what we sometimes call organization or cultural issues. And these are issues like getting people to share data. How do you handle IP? How do you give credit for team science in an environment of academia, which is really not terribly permissive for that? How do you train people in this relatively new area? What kind of novel collaborative structures do you need to make this happen? And we work on all of these. So the rare disease problem, the reason I show you this slide is not because you need to know it, because you know it already, but just because I like you to know how I talk about this. Every time I talk about this, I hear jaws hitting the floor because nobody knows this. And that's one of the reasons that we all need to keep up our education efforts, and Global Genes has been one of the real leaders in this. Something that you may not know is that there are currently about 250 new rare diseases identified every year, still. And what I mean by that, for those of you, the language will make sense. These are genotype-phenotype correlations. That is a relationship between a syndrome that a person has and a genetic defect, usually, that's been observed for the first time. It's really quite astounding number. So we haven't topped out on the number of rare diseases yet. One of the things that we're working on, and I'll just touch on this, is that rare diseases being rare, you almost always have to go international to either understand these or do clinical studies or natural history studies. And the definition of a rare disease varies according to countries, and so trying to harmonize that is something we're working on. But the bottom line is that, as you know, 95% of them have no treatment. So how are we organized in this way? Those of you who have been in the rare disease world for a while, that is more than 5 or 6 years, will know that the Office of Rare Diseases Research at NIH, which was originally in the Office of the Director, run by Steve Groft, who is just a giant in this field. He's officially retired, but still works for us. The directors now are Petra Kaufman, who's a neuromuscular person, and Ann Paris, who I've mentioned a couple times, who came to us from FDA in January. And we have a bunch of programs on rare diseases, and I'm not going to go through all these, but that's why I'm going to give you the links to them so that you can go and find more information. I'm just going to touch on a couple of them. The first, if you don't know about it, is GARD, which is the Genetic and Rare Disease Information Center. This is just what it sounds like. It's really intended for people who have just gotten a diagnosis. How do you find resources? How do you find people? Where do you find more information? What are the symptoms, et cetera? And there's actually a phone aspect of this because, as you all know, when you get a diagnosis like this, you want to talk to somebody who's really a doctor, and so there's a phone number as well, and so this has been a very active thing something that we do together with the Genome Institute, been doing for about 10 years now. One of the real centerpieces of the rare disease work that we do is this one, and some of you may be able to slot yourself into one of these. This is the Rare Disease Clinical Research Network, and is again a kind of complicated slide, but I just want you to take away a couple of points here. One is this thing I put in yellow here, and I'll highlight again here at the top, is that from the very beginning, this organization, this program has required the involvement of at least one patient group for the research team to get funding. So they have to be members of the research team in order to be funded by us in the first place. So there's a coalition of patient advocacy groups that's currently chaired by Chuck Mohan. Some of you may know, he's from the North American Mitochondrial Disease Foundation. And those little PAGs, those ovals, all of those, that means patient advocacy groups that are involved in each of these. So if the diseases that you work on fit into one of these, and there's about 200 diseases that this program works on now, that gives you an obvious place to link in. The other thing I want to point out here is that you notice if you look at the names of each of these, they're not individual diseases, and that's on purpose. So there are 7,000 diseases. There is no way that we can have a consortium on every single disease, and we think it actually wouldn't be the most efficient way to do it. These diseases are related to each other. It's one of the things that we found through the Genome Project and other things is that Mother Nature is actually much more networked than initially we thought, even 20 years ago. And so the same genes, the same pathways, et cetera, tend to get reused over and over and over again. That's a good thing for what we're all trying to do now, because it means that insights into one disease are more relevant than we might otherwise have thought of to others. And so what we ask these researchers to do is to say, well, tell us an organizing principle that you're going to use to study multiple diseases. And I'll pick on wherever the Alport syndrome person is, if she's still here. Ah, there she is. I'll pick on her again. It's a disorder of the kidney. And so rather than having a consortium on Alport syndrome, it would make most sense for that program to fit into, if I can find it here, the one on, where is it? Ah, here we go, Nephrotic Syndrome Study Network over here. So what that describes, nephrotic syndrome, is a syndrome of the kidneys, and it can be caused by many different things. And so this network studies that group of diseases in order to, so that when a finding is found in one, it can be translated rapidly into another. So you notice some people have organized it around syndromes, disease syndromes like this. Some people have organized around an organelle, like the lysosome. You've heard about lysosomal storage diseases or mitochondria. Some people have organized around a cell type. So you see this one, where is it? The inherited neuropathies. These are peripheral nerves. And some people organize around a whole organ, rare lung diseases. So the point is that we're organizing research in a different way, and we're organizing the teams in a different way to make this much more efficient. I mentioned the toolkit, and I was going to give you a tour here, but I hope they've sent you the link, which is on the bottom here. I sent it to the organizers, and they should be adding it to the link on the page of the session we just had with John. And so go take a look. It is a really cool site, if I do say so myself. It really is quite wonderful. And you'll notice what it says. I'll just read you what it says. The toolkit was developed to provide your patient group with the tools needed to advance medical research. And you notice the areas at the top are, getting started. Where do you start? The discovery phase, that is going from a gene to something that you might want to put into a clinical trial, preparing for a clinical trial, clinical trials and the FDA, after FDA approval, things like access. And then under each of those, there are tools that are there for you. So I'm just going to just took us through screenshots here. If you click on Getting Started, this is what it looks like, just as an example. Learning about therapeutic development. How is a therapy developed? Why is it important for patients to be involved? How do I begin? Building relationships, all of that. And if you click, in this case on this one, how is a therapy developed? These are a bunch of tools here, and you'll notice this one comes from Global Genes, but they come from many places. The one above it comes from the FDA, and they come from all over the place, added to this toolkit. So this is really a quite comprehensive toolkit that we hope you'll really find helpful. And it's been written, it's not written in what I affectionately like to call geek speak. It's not written by and for scientists, because we speak this weird idiosyncratic language nobody else, including my wife, seems to understand. And so we wrote it for folks who are not specialists in science. Okay, so now I am going to give you some information that is going to throw some science at you, and I do it on purpose, which is to say that we do have a lot of hardcore scientists involved with our place, but we view this process a lot like using an iPhone. I have no idea how an iPhone works. I don't want to know how an iPhone works. I want to trust that Apple has really smart, geeky people who are making my life easier, but I don't want to know about it. I just want to know the bottom line and how to use it, make it easy. And that's how this whole process should work for you. But I did want to give you a sense of what it looks like under the hood so that you can be assured that, that these are good people and how the whole process works. Okay, so this was something that I mentioned briefly in our session with John. This is one of many, many, many projects I just picked out to talk to you about. It's a collaborative project to develop gene therapy. So we've, along with a lot of other folks, have gotten interested in the renaissance of gene therapy in the last 5 years due to some real important advances in the basic biology of gene therapy. And it's allowed us to begin to think about gene therapy as a really viable option for patients again. But it's not going to happen by itself. It's certainly not going to happen that way in a generalizable way. So we have a number of projects that we've taken on now within this Therapeutics for Rare Neglected Diseases program, or TREN, to try out different aspects of the gene therapy problem and that will then be disseminatable, that will be applicable to other disorders. As well as hopefully developing a therapy for an individual disease. So again, like everything we do at NCAS is a collaboration. It happens to be a collaboration between a company called Agilus Therapeutics, it's based in Boston, the TREN program, and clinical investigators at the National University in Taiwan. The disease is AADC deficiency. The desired intervention here is a single dose of what's called AAVH-AADC. What does that mean? That's an adeno-associated vector, adeno-associated virus. That's a virus that's carrying the human AADC gene into the brain, into the part of the brain that's affected in this disorder. The challenges are things that some of you are probably familiar with. It's an ultra-rare disease. It's almost certainly underdiagnosed. It requires stereotactic surgery in newborns or infant brains and all kinds of CMC regulatory issues. And so we teamed up with them to solve these problems. And all of the words at the bottom there, TREN collaboration that started about 1.5 years ago, was doing all the work that's required to make this product potentially acceptable to the FDA. And we had a really important regulatory meeting with FDA over the summer that means that the BLA, that is the application, might go in 2018. And importantly, this is currently the most advanced brain gene therapy in development. But the other important thing here, there are two important things here. One is the potential clinical effect, which I'm going to show you in a second, which really motivated us to take the risks on this disorder, but also because for a variety of reasons that are diagrammed in all the brain pictures and biochemical pathways on the right, there's good reason to believe that if we're successful here, these will create precedents which will be useful to us and to the FDA for approving other therapies, because that's the whole idea here. We want every treatment or every project to be a dual-use project, right? Moves a project down the translational pipeline, but also teaches us something general. Okay, so here's the disease, and I just had to show you this because this will illustrate why we decided to take this on. So this is a child who's, at this age, she's 4 years old. She's in Taiwan, where this study was done. And you'll notice that the child has no independent spontaneous motor movement, has a little bit of responsiveness in her face, but very little activity, some of it stereotyped activity. At this point, she got the gene therapy that is now under study, and this is her now a year later. Now it is formally possible that this happened by itself. You know, it's a single case. It's possible. It's never been seen in this disorder before. Extraordinarily unlikely, but of course that's why clinical trials are necessary, to make sure that this is not a spontaneous remission. But it is almost certainly not. And now here's her 2 years after the gene therapy. So this is a single dose of the gene therapy. And if I showed you this on the right, and I asked you, you can't hear the audio, but she's speaking to her mother too. You would be, oh, there she is. You would be hard pressed. Now, especially if you know Chinese, you would be hard pressed to say that this child is not totally normal. So that's the question. The question is how to take potential, like what's like this, and how to make it real. And the point I want to make to you is that this really is possible these days. When I was in training 30 years ago, it was not possible. But now there are organizations like Global Genes and NCATS and others that exist to work with you to make these things happen. Okay, so I'm going to move backwards a little bit. So that's a program that you should look into if you're at that stage. Most projects are not at that stage. They frequently have a gene or have understanding of a disorder, but they don't have even a start of an intervention in this case being a drug or a device or an antibody or a protein or a gene therapy, something that you're going to give to the patient. And so what do you do in this case? Well, this is one program within another program within NCATS that you should consider. It's something called the ADST Laboratory, but the point is that this lab works in collaborative relationships with disease foundations to, as it says, enable drug development for early stage, the first steps out of an academic lab to get to the process of therapeutic development. We also do a lot of training and support for researchers in the foundations that fund these researchers really like that too, because it builds their capacity for their disease as well. And I don't know if Jim's here from the Cure GM Foundation. I thought he might be here, but there he is. And you see the juvenile dermatomyositis group here at the bottom. But there's Alpha-1 is here someplace too. There she is. Alpha-1's in the second row. CMTA, many others. And it's really turned out to be a very potent model, bringing together patient advocacy groups, academic researchers, and the translational experts at NCATS. And it really is a three-legged stool. That's the only reason it works, is that you got to have all three or else the programs fail. Important thing I just want to emphasize again and again and again, and I'm sorry to be making the same point over again, but I think it really is important that we do a lot of patient-driven science. And these are just 3 papers, this one on a peripheral neuropathy known as Charcot-Marie-Tooth, this one on a rare cancer called chordoma, and this is on a rare a myopathy called myotonic dystrophy, where each of these projects started because a parent or a patient walked into my office and said, "Can we work with you?" And so this is something we're very committed to as a model because it works. It really does work. Okay, so last 2 things I want to tell you about, and then we can have some discussion, I hope. This is something that you probably can't interface with now, but I want you to know about it because it's another technology that I think is going to be absolutely transformative in the work that we're all trying to do. The goal here is to develop what are called microphysiological systems, if you hear that term in a scientific terminology, otherwise known as tissue chips. Sometimes they're known as organs on chips. Although I've gotten away from calling it that because once when I said I was talking about a liver chip, and somebody said, "Wait, are you talking about pate?" That's a liver chip. No, it's a liver on a chip is not pate. No, this is again translational problem. So these are 3-dimensional constructs of human cells, multicellular aggregates of human cells that represent the structure and function of a human tissue and are kept alive for months at a time and can be used potentially as a way for testing the safety and efficacy of drugs. So it's a long, really interesting story of the convergence of informatics and engineering and biology. But the important thing is that over the last 5 years, this real space age idea that really sort of takes us back to Star Trek is turning out to be much more possible and has happened much more rapidly than many of us anticipated. So if you go to our website, you can actually find this gentleman in the upper right here. He's actually not a gentleman because he has both sex organs. He's actually a hermaphrodite. But if you click, his name is Chip. Yes, sorry. And if you click on any of Chip's organs here, so you can see Chip's the tissue chip systems which have been developed. The point I want to make here beyond this is this. So again, a kind of a geeky slide, but I want to draw your attention to the part that's moving. So in the midst of developing a heart chip, and by the way, I should make it clear, why do we call these chips? It's not, the analogy is not to potato chips. The analogy is to computer chips. But instead of electrons moving around in a computer chip, it's a liquid moving around in a microfluidic system. That's why we call them chips. Okay, so the heart chip was made from induced pluripotent stem cells that were differentiated into cardiomyocytes, heart cells, and they start spontaneously beating. That's what you're seeing on the left, on the lower left under D. That's the heart muscle doing its sort of sit-ups that heart muscle does, even spontaneously. Now, it turned out that the people who were developing this, who happened to be up at Harvard at the Wyss Institute, there were some people down the street, down the hall, in a lab studying a rare disorder called Barth syndrome. And it's a multi-system disorder, a rare disorder. And these kids die of a congestive heart failure, dilated congestive heart failure, and it wasn't clear why. And so they thought, well, gosh, if we can make a normal heart chip out of a heart chip that contracts normally from the stem cells of normal people, couldn't we take iPS cells from children with Barth syndrome and make a model of the congestive heart failure that happens in Barth syndrome? So that's exactly what they did. And what you're looking at in the middle, the one that's barely moving at all, that's a heart chip made from cells from Barth syndrome children. And they don't contract, just like you see in the kids. The one on the right is if you take those Barth syndrome cells, you insert the normal gene, it's called tafazin, it's the gene that's mutated in Barth syndrome, and it fixes the defect. So in other words, it cures the disease on a chip. So what this does is it created, all of a sudden, a model for Barth syndrome, a way to test therapies for Barth syndrome, which we never had before. Well, this is the first example of this, but we've now seen it in many other organs. So we've actually started a program, and there's lots of papers. If you ever want to read the paper, there it is. But got all sorts of awards and things. But the important thing is that then we said, okay, we're going to now have a program in stage 2 of this tissue chip initiative, a specific program to ask disease researchers, mainly rare disease researchers, to come in and we're going to support them to develop models of rare diseases in these tissue chip systems. And so that's something that is just happening now. It's really leading edge technology. It's just happening now, but I wanted you to know about it because you're going to be hearing a lot more about this. I think in the next few years, there are companies being formed, spun out to commercialize these technologies and democratize the technologies. So I hope you're going to hear more about it, and I'm quite confident you will. The last thing I just want to touch on, and again, this is something that you need to know about, and Global Genes will certainly be involved in this as an umbrella group. But I want to make sure you know about it. Because I hope it will give you more hope that we're finally getting our act together as a community to address this problem in a global way. And by global, I mean geographic global and global in terms of all diseases. So this is something called the International Rare Diseases Research Consortium. I became chair of this organization about 1.5 years ago. What is this? This is a global consortium of government funders from 20 countries and 5 continents that now also has many biotech companies and pharma companies involved, many umbrella patient advocacy groups from the various countries, and scientists who are coordinating their research to make more headway across all the diseases much more rapidly by coordination and cooperation. I just want to finish my last slide here. In addition to, I'm going to show you the contact points. I want to I want to tell you what this group committed itself to just last month. So this is on the left is an article in Nature that I and my co-chair Hugh Dawkins in Australia wrote, and a bunch of papers that we published in the scientific literature about this too. But I want you to focus what's on the right side. So again, this was put together by government funders, nonprofits, patient groups, companies, scientists, and we asked ourselves, well, we want to challenge ourselves to goals for the next decade that are outlandish enough to make people change their behavior, so they're not achievable now, but are not totally impractical, because that's just not helpful. And this is what we came up with. We came up with a vision. The vision, long-term vision, is to enable all people living with a rare disease to receive diagnosis, care, and therapy, and this is really where the zinger comes in, within 1 year of coming to subspecialty medical attention. That, is, all of you know, does not happen now, but there's no reason it couldn't. This is not limited by the laws of physics. It's limited by human-created systems. So to support the vision over the next decade, what have we committed ourselves to achieve? That the first goal is diagnostic, that all patients coming to medical attention with a suspected rare disease will be diagnosed within 1 year of coming to diagnosis, or coming to subspecialty medical attention, if the disorder has previously been defined. Right? So if it's alpha-1 or it's Alport syndrome or it's juvenile dermatomyositis, that has been described in the literature. There is no reason that those children should not be efficiently diagnosed. But there are many diseases which still remain undiagnosable. And so this organization has committed itself to a globally coordinated diagnostic pipeline to diagnose those people. Goal 2 is that by the end of the next decade, there will be 1,000 new therapies for rare diseases that have been approved. And this is important on the second one, the majority of which will focus on diseases with no therapy. So if we all celebrate the findings or the developments in cystic fibrosis, for instance, really led the way. But what this organization really is focused on is not cystic fibrosis, but on all the other disorders which would like to be cystic fibrosis. And the third goal is, I think you'll find interesting too. We spent a lot of time talking about well, we're a research organization. How do we think about the issue of access? That is, that if a therapy is approved but nobody gets it, or people get it but they don't improve, have we succeeded or not? And I'm glad to tell you that the group decided, as a group, that the answer is no. Now, we're a research organization. We're not a funder. We're not an insurance company. We're not a health technology assessment organization. But we find that many of the problems associated with reimbursement and pricing and all that stuff come from inadequate methodologies. It's a scientific problem that the folks who are tasked with doing this just don't have the data they need to make decisions. And so ERDR has committed itself to developing methodologies that will assess the impact of diagnoses and therapies on the lives of rare disease patients. Okay, so you'll have this, and you should have this in your app. But these are the links to and people that you should email with interest in any of these programs that I mentioned. The only one I didn't mention by name is the NIH Chemical Genomics Center. That is a much bigger version of the ADST group. The ADST group is one specific one, but the NCGC is about 10 times that size, works on many common diseases as well. So with that, I am going to finish. If you want to connect with us and be kept up to date on what we do, the easiest thing to do is just to go to this Connect part of our website, and you can sign up, and you'll get our newsletters. You'll get my ever popular director's message that comes out every month. I think you'd actually find it kind of interesting in terms of what we're doing and how we think about translational science. And I think at this point, if we have time, I hope we do, I'd love to take questions, have whatever's on your mind, and be glad to talk to you about what we're doing and what you think we ought to be doing that we're not already doing, or et cetera, et cetera, et cetera. So, do you want to?
Thank you, Doctor. My name's Eric Cartman. I'm with the Choroideremia Research Foundation. It's a rare inherited disease. And I also want to thank NCATS for that Rare Disease Day that you guys sponsored.
Oh, yeah.
That was a great event,
Yeah,
and it was a
thank
great
you.
intro
Yeah.
for many of us. And I look forward to the next one.
Yeah, great.
My disease, and it's part of what is luckily a growing trend within inherited rare eye diseases, is that we've got a lot of different gene therapy trials that are either, I've actually got 2 different trials underway right now.
Yep.
The problem we have is supposedly our eye disease has 1 in 50,000, so 6,000 in the United States. My foundation's been around 17 years. We've only found 500 people. And the vast majority, 95% of our members, were misdiagnosed as RP. And given the current situation of these multiple RP and other eye disorders, how do we mount a genotyping program?
Yes. Oh, boy. Now you said the magic word. Yeah, keep going.
Well, because without that, we're doing everything to Ancestry.com and trying to find family strains that go off, and suddenly we're finding a whole nother group of relatives of someone that have been living with an RP diagnosis, and they now know they have choroideremia, need to get genotyped. And it's a question of getting that word out there. And I would think getting the health providers to begin to understand that genotyping is important, especially where there are therapies that are beginning to be developed or are being developed.
What a fantastic question. And I hope you in case you didn't understand how important that question was for the most part, rare diseases are still diagnosed on a syndromic basis, that is clinical diagnosis. And clinicians, as good as they are, there's many ways to get to an endpoint that looks the same in the clinic. And so what tends to happen is people when a physician sees something in the clinic, they not unreasonably say, well, odds are that it's this. In this case, it's the most common one, which is retinitis pigmentosa. But it could be a whole host of other disorders. Now, 20 years ago, 30 years ago, there wasn't anything we could do. We had no other way to figure this out. But now, for many disorders, rare diseases, there are, of course, genotypes. There's a way to do a sequencing of the relevant genes or the entire genome and make a definitive diagnosis in a way that was never possible before. This, to me, is the obvious answer to this entire rare disease diagnostic problem. But it has not moved forward for a very simple reason, and you hit on it, and I think all of you as patient advocates could really help with this, which is that this technology, that is genome sequencing, of course, was invented for a very basic reason. That is, it was to do the Genome Project, and I showed you that before. But now it has transitioned into what is now more frequently called medical sequencing. That is, that you're sequencing a person to make a diagnosis. But for the most part, insurers think of this technology still as a basic experimental technology, and so they won't reimburse for it. And it results in problems that we see in our healthcare system in other ways, which is, you think about how these folks are thinking, that they're trying to minimize cost in the short term, and the long-term effects are less important to them. Whereas we go to them and we say, and we're doing this a lot through ERDIC, is to say, look, if you made this diagnosis today, you could prevent a diagnostic odyssey, would prevent these parents or people from having to go see 15 other doctors, and they're going to get the same test done over and over again. Huge healthcare expenses. They will spin their wheels for the next 15 years and finally get a diagnosis. That's very costly. Meanwhile, the patient is deteriorating. And that's very costly. So wouldn't it make sense to do a relatively cheap intervention? Genome sequencing is now, whole genome sequencing is about $3,000. It's about the cost of an MRI. To do this once, and one would have the diagnosis. But so far, and this is turning out to be a health technology, it's a health economics question. That when we talk to the insurers, they say, sure, we take your point that that might be true, but we need data to show us that it really is going to save money. And that's why ERDR particularly, but also our place, are focused on this question. This is particularly an issue, I should say, in kids, in newborns. There's a really groundbreaking effort that if you haven't read about, you really should read about it at not far from here at Rady Children's in San Diego, where they take children in the NICU, in the neonatal ICU, who are really sick and they have no idea what's going on with them. Within 5 days, they can go from a genome to a diagnosis in about half the kids and literally save their lives. So I think this is another change in the way our health system needs to develop. And we can talk about this from our perspective, and we do, but you're the consumers, and they're going to listen to you.
Okay. Hi, my name is Mary. It's good to hear you. I'm glad when you said, and the cost of a new drug goes to infinity.
Yeah.
That's, in my experience, I have a lamin A mutation that causes limb-girdle, muscular dystrophy, and cardiac cardiomyopathy, conduction block.
Yep.
And anyway, I did join a clinical trial. I didn't meet the inclusion criteria, but the document, the informed consent document, was a nightmare, and it was just, I asked a few too many questions, and I could tell they were getting pissed off at me, and so that wasn't a good experience, and so I hesitate to join any other trial because I feel that if a therapy comes to fruition that works, patients will be priced out of it.
Yeah, right.
And sure, it's just there has to be a cap.
Yeah.
If tax dollars are being used to research and make these drugs, then there's got to be a cap. You can't have a drug that works and patients can't get it.
Yeah.
So I don't know, it's not a happy comment, but that's where I'm at. That's where my heart is at today. Thanks.
Well, it's a really important point, and it's something that we struggle with all the time because, of course, as a research agency, we don't have any direct impact, nor should we, because it's not the purpose of our agency. However, we can't be blind to it. I mean, if all the work that we do, if patients don't get access, why are we bothering? So we do enter into this discussion a lot, and you should know we get asked about this question a lot by policymakers in Washington. And it's a complicated subject, as you know, but we're approaching it at least two ways. One is that some of the problem here is that drugs which are marginally effective or not even very effective, or are old drugs where their development was done decades ago, have very high prices, and there's just no reason for that. There's no way you could say that we're recouping development costs. It's just absurd. And so to have some way to affect that seems pretty obvious. The other, though, is that and really we think the long-term solution here is to reverse that Eroom's law graph that I showed you. Because as long as that happens, as long as efficiency decreases progressively, then we're never going to be able to solve this problem. We can put our finger in the dike, but ultimately what we need to be able to do is to turn that graph to go up again. So how do we make efficiency improve? If we do that, then the cost of production will come down. That will allow prices to come down. Doesn't necessarily mean they will, but if cost of production comes down, then that allows prices to go down, because you will be able, it'll work like an iPhone does, right? So, or not the most recent iPhone, because I guess the price went back up again, but for the most part, you know, the cost of televisions or whatever it is, you know, they tend to come down over time, and that hasn't happened. But the reason is that this efficiency has not increased, and that's a research question. At the same time, we focus a lot on that, how to, and when you think about this, John and I were talking about this before, you know, why are drugs so expensive? It's not so much the unit cost of production, it's the fact that the failure rate is so high that for every success, you got to do, you know, 9, 10, you know, projects for everyone that succeeds. So if you decrease the failure rate, that's going to have a much better effect, much more larger effect on the cost. Now, the last thing, however, which is really important to this, and I want to come back to this, which is the health technology assessment issue. That is, if you had a therapy like what you're describing, and if the payers have no way to determine whether this therapy actually had a beneficial effect on patients, they will not pay for it. And we actually saw this happen in Europe. I don't know if you followed this, but the first gene therapy ever approved in Europe, something called Libera for lipoprotein lipase deficiency, was $1 million per dose, and it was taken off the market earlier this year because it didn't sell. It just was too expensive and people wouldn't pay for it. And so what you're describing is real. It's not a matter of only our patients not going to be able to get it, but companies aren't going to, they're going to stop making these things. So one of the things that our system has to come up with is a much more robust way to assess the effects of these on real patients' lives and distinguish between ones that really work and ones that don't work. And currently, we're paying high prices for almost everything, and that's some of the problem. So it's not a great answer. I'm not sure there is an answer, but anyway, go ahead. Yeah. Yes. Right. Yep. Yeah. And this is a great point. So the PCSK9, these are, you probably know what these are. They're for cholesterol. It's a great example of what's happening. That because because it's been hard so far to distinguish between drugs that really work, like even if they're expensive, like PCSK9 or the Hep C drugs is another example. You know, they're curative, prevent liver transplants, quite amazing. But insurers don't want to pay for that either because they're paying for all this other stuff. And that's the question. How do we distinguish ones that work for what's done, if what don't? And then the insurers are fine with paying things that actually work if they have data. But currently they can't distinguish those, and that's the problem. And so they just cut everything. And that's just not, it's just not, it's not an optimal solution, put it that way. So yeah, we got to get done. Yeah.
But thank you so much for all that great information. And if everyone could give Chris a round of applause. Thanks. We ran a good bit over, so it's going to be pretty tight for you guys to get to your next session. But thank you for joining us, and we will see you this afternoon back in here for our awards.